Katie Doores. Katherine Jane Doores is a British biochemist who is a senior lecturer in the School of Immunology and Microbial Sciences at King's College London. During the COVID-19 pandemic, Doores studied the levels of antibodies in patients who had suffered from coronavirus disease. Early life and education. Doores was born in the United Kingdom. In 2003, Doores received an MChem in chemistry from the University of Oxford. In 2008, Doores received a PhD in organic chemistry from the University of Oxford. N.G. Davis was her advisor. In 2013, she completed postdoctoral work in the Department of Immunology and Microbial Sciences at the Scripps Research Institute in La Jolla, California. Research and career. When Doores was a graduate student at Oxford, she studied glycoimmunology in the laboratory of Professor Davis. Glycoimmunology is an emerging research field which looks at how immune response is moderated by carbohydrates glycans. At the Scripps Research Institute, as part of her postdoctoral work, she worked on the glycobiology on HIV and broadly neutralizing antibodies. At Scripps, Doores worked alongside Dennis Burton, where she studied the flower-like envelope protein on HIV. These envelope protein penetrates host cells and create antibody-resistant glycans. By investigating this envelope protein, Doores looked to identify sites which are involved with viral function. By neutralizing sites such as these, the high mannose patch, Doores hoped to protect against HIV infection. From 2013 to 2017, Doores was a lecturer in the Department of Infectious Diseases at King's College London. Doores was awarded a Medical Research Council Fellowship to establish her own laboratory at King's College. She was made a European Molecular Biology Organization EMBO Young Investigator in 2017. In 2017, Doores became a senior lecturer in the Department of Infectious Diseases at King's College. Many disease-causing pathogens are coated in carbohydrates. Doores investigates the behavior of these carbohydrates in host pathogen interactions. She hopes that by understanding the role of these carbohydrates, it will be possible to develop novel therapeutic strategies and vaccinations. Alongside developing new medical therapies, Doores is interested in how the body responds to carbohydrate antigens in the form of antibody recognition. Her work has primarily focused on the carbohydrate antigens on HIV-1. The envelope glycoprotein GP120 of HIV-1 is covered in and linked glycans. These glycans are the target of bNAb's broadly neutralizing HIV-1 antibodies, and Doores is studying how these antibodies evolve in vivo. This understanding will allow the development of new vaccines that encourage the generation of antibodies that can protect against pathogenic bacteria. During the COVID-19 pandemic, Doores studied the levels of antibodies in patients who had suffered from coronavirus disease in Guy's and St. Thomas NHS Foundation Trust. Her research showed that while 60% of coronavirus disease patients elicited a strong antibody response, only 17% of them retained this potency 3 months later. In some cases, patients entirely lost their antibody response. These results implied that immunity to coronavirus disease might be short-lived and that people may become reinfected during a second wave of infection. Selected publications. Doores, Katie J. 2007. Novel Methods for the Synthesis of Glycoimmunological Probes, Ph.D. University of Oxford. Mathematical and Physical Sciences Division. OCLC 232,193,273. Doores, K.J. Abonomally, C. Harvey, D.J. Vasiljevic, S. Jevic, S. Dweck, R.A. Burton, D.R. Crispin, M. Scanlon, C.N. 19 July 2010. Envelope lichens of immunodeficiency virions are almost entirely oligomannose antigens, PDF. Proceedings of the National Academy of Sciences. 107-31-13813,805. doi 10.1073/PNAS 1,006,498,107, PMC 2,922,250, PMID 20,643,940, Wikidata Doores, Katie J. Burton, Dennis R. 15 October 2010. Variable loop glycan dependency of the broad and potent HIV-1 neutralizing antibodies P9 and PG16. Journal of Virology. 8420, 10,510, 10,591. doi 10.1128/JVI 552-10. PMC 2,950,566. PMID 20,686,044. Wikidata Pedgel R. Doores KJ. Walker LM. Kayat R. Huang P. S. Wang S. K. Stanfield R.L. Julian J. P. Ramos A. Crispin M. Petrus R. Capelli U. Marason A. Cupo A. Malavest S. Liu Y. McBride R. Ito Y. Sanders R. W. Agohara C. Paulson J. C. Feezy T. Scanlon C. N. Wang C. H. Moore J. P. Olson W. C. Ward A. B. Poignard P. Sheaf W. R. Burton D. R. Wilson I. A. 13 October 2011. A potent and broad neutralizing antibody recognizes and penetrates the HIV glycan shield. Science. 334 6059, 1097 1103. doi 10.1126/science 1,213,256. PMC 3,280,215. PMID 21,998,254. Wikidata Walker Laura M. Huber Michael Doores Katie J. Falkowska Emilia Pedgel Robert Julian Jean Philip Wang Shankai Ramos Alejandra Chan Huey Po Ying Moll Matthew Mitcham Jennifer L. Hammond Philip W. Olson Ol A. Fung Pam Fling Stephen Wang Chai Huey Foggett Sanjay Rin Terry Simic Melissa D. Principal Investigators Protocol G. Koff Wayne C. Wilson Ian A. Burton Dennis R. Poignard Pascal 22 September 2011. Broad neutralization coverage of HIV by multiple highly potent antibodies. Nature. 477 7365, 466 470. doi 10.1038/Nature 373. PMC 3,393,110. PMID 21,849,977. Wikidata Soc D Lock D Yuck D Doores KJ Briney B Le KM Say Francisco KL Ramos A Culp DW Julian J P Menes S Wickremessing L Seaman MS Sheaf W R Wilson I A Poignard P. Burton D. R. 14 May 2014. Promiscuous glycan site recognition by antibodies to the Hymanos patch of GPON 120 broadens neutralization of HIV. Science Translational Medicine. 6, 236. 236 or 63. doi 10.1126/Satransl 3,008,104, PMC 4,095,976, PMID 24,828,077, Wikidata, Kidata, Seabright, Gemma E. Doores, Katie J. Burton, Dennis R. Crispin, Max, May 2019. Protein and Glycan Mimicry in HIV Vaccine Design. Journal of Molecular Biology. 431 12 22 23 22 47. doi 10.1016/JJMB 2019.4 16. PMC 6,556,556. PMID 31,028,779. Wikidata, Kidata. Avanzato, Victoria A. Agantuyo, Casofalu Y. Escalorazamudio, Marina, Gutierrez, Bernardo, Golden, Michael, Kosakovsky Pond, Sergey L. Price, Reese, Walter, Thomas S. C.O. Jeffrey, Doores, Katie J. Pybus, Oliver G. Munster, Vincent J. Lee, Benher, Bowden, Thomas A. 10 December 2019. A structural basis for antibody-mediated neutralization of Nipah virus reveals a site of vulnerability at the fusion glycoprotein apex PDF. Proceedings of the National Academy of Sciences. 11650, 25,057, 25,067. doi 10.1073/PNAS 1,912,503,116. PMC 6,911,215. PMID 31,767,754. Wikidata, Kidata. C.O. Jeffrey, Graham Carl, Merrick, Blair, Ackers, Sam Steele, Katherine J. Hemmings, Oliver, O'Brien, Aoife, Khufu, Neophytos, Pickering, Susan, L. Rui, Betancourt, Gilberto, Wilson, Harry D. Signell, Adrian W. Winstone, Helena, Carriage, Claire, Neil Stewart, J. D. Mullum, Michael H. Doores, Katie 11 July 2020. Longitudinal Evaluation and Decline of Antibody Responses in SARS-CoV-2 Infection PDF. Med Arsif preprint. doi 10.1101/2020.7. 9.20,148,429. SDU CID 220,480,492.